Today we're going to talk about the cell cycle and cell growth and division. Biology is the only subject in which multiplication is the same thing as division. So where it all began, you started as a cell smaller than a period at the end of a sentence, and now look at you. So how did we get from there to here? Getting from there to here, we're going from the egg to a baby. The original fertilized egg has to divide and divide and divide and divide. So you start as an ovary in your mom, and when it's fertilized by a sperm, it generates and starts the rapid cell division, which will eventually lead to a baby and ultimately to you, a human. So what is the purpose of cell division? For reproduction, for growth, repair and renewal, and to limit cell size. So for reproduction, to create offspring, there's also asexual reproduction, which is a single cell organism reproducing on its own, so it creates an identical clone. For growth and organization, or growth of an organism, sorry. So when you start off with that fertilized egg, you are growing into a human, but you have to start off as a single cell, and then you multiply and divide and multiply and divide to form the multicellular human. For repair and renewal, so in the case of the starfish, if the starfish limbs are pulled off, it will regenerate its legs. Or in the case of a lizard, if it's trying to get away from a predator, it can detach from its tail and it needs to regrow its tail, and to limit cell size. So making new cells, there are different parts that we have to worry about. So we have our nucleus, which we know is the house for our DNA. Our DNA is wound up on chromosomes. We have our cytoskeleton, which is made up of centromeres and centrioles in animals, and spindle fibers. So we have our cytoskeleton, which is made up of our centromeres and centrioles in animals. So those are up here, as well as the spindle fibers. So before dividing, what has to be copied? You need to copy your DNA, your organelles, your cell membrane, lots of other molecules like proteins and enzymes, because you're trying to make two exact copies. And if you don't have all of the proper material, you only have half, you're not going to produce two identical daughter cells. So the cell cycle equals the life cycle of the cell. There's something called interphase, which is where the cell spends most of its lifetime. So it's 90% of the cell's life cycle. And with the interphase, there are three phases. There's the G1, the S, and G2. G1 and G2 are growth phases, and the S phase is DNA synthesis. So it's the copying and replication of DNA. So this is what the phase in which cells are just doing and performing their everyday tasks. They're producing RNA, they're synthesizing proteins and enzymes. So during M phase, that's mitosis and cytokinesis. Mitosis is known as the nuclear division, so that's when the DNA divides, and cytokinesis is when the cell divides. So cyto meaning cell, kinesis meaning movement, so it's the movement of the cells. So interphase is divided into three phases. The G1 is the first gap or growth, and this is when the cell is doing its everyday job. It can go to G0. G0 is for cells that don't need to replicate or duplicate in any way, shape, or form. So G0 is the phase where your nerve cells and muscle cells typically are, as well as your liver cells, because your liver cells only reproduce every couple of years, so it goes into this G0 phase. And the cell grows, and there needs to be a signal for division. So when there's a signal for division, or signal to divide, we'll start the S phase and DNA synthesis will begin. So the chromosomes are copied. Once chromosomes are copied, we will go into our G2 phase, or the second gap growth phase, and this is preparation for division. The cell continues to grow to make sure everything is there, the organelles, the proteins, and the membranes. So in interphase, the nucleus is well defined. The DNA is loosely packed in long chromatin fibers. The nucleolus is visible, and it's preparing for the M phase. So in preparation for the M phase, it's replicating chromosomes or copying the DNA and proteins. It's producing proteins and organelles, and it's duplicating its centrosomes. So during this phase, the DNA is loosely packed because it doesn't need to divide. When it's loosely packed like this, it's also not a good idea to pull it apart because you'll tangle up your DNA if the cell divides at this point and probably ruin the DNA. So S phase, or the synthesis phase, is the copying or replicating of DNA. This is when DNA replication occurs, and it will be part of dividing the cells. The dividing cell will replicate its DNA. So during S phase, the cell is getting ready to divide. It's in preparation, so it needs 2 sets of the DNA. Because if you don't copy the DNA, the daughter cells are only going to get half the DNA, and that's not what the cell needs. It needs all of the DNA, so it has all of the information or genetic material it's going to need to decide what it's going to become. So before replication, and then after replication. So during the S phase, we've seen this because we've talked about DNA replication. We have our DNA. It begins to unwind and unzip from helicase. The DNA polymerase comes in and copies the DNA and will result in 2 daughter strands that are connected by the centromere. So copying DNA and packaging it after duplication, the chromatin begins to condense. So the coiling and folding to make a smaller package. So all this loosely tangled DNA is now condensing down, wrapping around its histone proteins to form nice chromosomes. So organizing DNA. DNA is organized in chromosomes. The double helix DNA molecule is wrapped around histone proteins, like the spool around the thread. The DNA protein complex is called the nucleosome. It's organized in long, thin fibers and will condense further during mitosis. So the picture on the right is just showing you the DNA double helix will wrap around the histone proteins and condense down and make this little chromatin fiber and coil up even further to make your chromosomes. So it's a double-stranded chromosome which is connected by the centromere. So mitotic chromosomes are made up of 2 sister chromatids and a centromere. When these separate, this is one chromosome, but when they separate, they will end up being individual chromosomes. So here we have one chromatid. So duplicated chromosome, you have 2 sister chromatids narrowed at the centromere, and they contain identical copies of the original DNA. So during S phase, this single chromatid will undergo DNA replication and chromosome duplication, and you'll end up with sister chromatids. So these are pairs of double-stranded mitotic human chromosomes, which we've looked at before. So now that the DNA is replicated, we're ready for the process of mitosis. Mitosis is the dividing of cells' DNA between two daughter nuclei. So it's the dance of the chromosomes. And there are 4 phases. You can remember as PMAT, so you've got prophase, metaphase, anaphase, telophase. So if you want just a basic overview, we start in the interphase with the loosely condensed chromatin. Then we've got early prophase, late prophase, metaphase, anaphase, telophase. Telophase is the final phase when the nuclear membrane begins to form again, and there starts to be a cleaving. And cytokinesis is finally when the cells start to separate into their individual cells. During metaphase, the chromosomes will line up on something called the metaphase plate, which is the center of the cell, so that the chromosomes can be pulled to the opposite poles. So prophase, the chromatin condenses, the centrosomes move to opposite poles of the cell, or the centrosomes or the centrioles in animal cells. The mitotic spindles form and attach to the centromere of the chromosomes. The nucleolus disappears and the nuclear membrane breaks down. So the cell in prophase is getting ready to get the chromosomes, or the chromosomes lined up on the center so that they can divide. The spindle fibers need to be attached, or the mitotic spindles need to be attached, so that the chromosomes can be pulled to the opposite poles. During metaphase, like I said, the chromosomes are going to line up along the metaphase plate. So they're going to line on the equator of the cell, called the metaphase plate, meta meaning middle, and get ready to be pulled apart. So if we look at these cells, this is kind of what it looks like under the microscope. You have the chromosomes all lined up in the center of the cell. In anaphase, the sister chromatids separate, so they move to the opposite poles and are pulled at the centromeres and pulled by the spindle fibers. So if the spindle fibers had not connected to the centromeres and the centrioles weren't attached to the spindle fibers, the sister chromatids would be stuck at the metaphase plate. So they begin to move, they're getting pulled, and the poles move farther apart, so the spindle fibers lengthen. And then we get into telophase. Telophase is the chromosomes are finished traveling to the poles, so they're officially there. The nuclear envelope begins to reappear, and the daughter nuclei is formed. And the nucleoli is reforming. The chromosomes begin to uncoil and are no longer visible, and they're back to chromatin form. The spindle fibers disappear, and cytokinesis begins, which is the cell division. So cytokinesis in animals is division from the outside in. It's called the cleavage furrow. The cells will begin to start pinching off, and that's called the cleavage furrow, and splits into the cells split into 2, and it's like tightening the drawstring. So they start pinching in on the sides and keep pulling until you finally end up with the 2 cells. Cytokinesis in plants is a little different. The division is from inside out. So the plant cells form a cell plate. The vesicles line up at the equator, and the vesicles fuse to form 2 cell membranes. The new cell wall laid down between the membranes, and the new cells fuse with existing cell wall. So it'll start off as little vesicles containing cell wall material. The cell plate will form, and then you will develop the two daughter cells. So that's cytokinesis in a plant cell. You can see the little vesicles forming along the equator. And this is what mitosis looks in a white fish blastula. So you have your interphase where the chromatin is just, you can see the nucleus in the chromatin. You can see prophase where the chromatin is starting to condense. Metaphase, the chromatids are lined up at the metaphase plate. Anaphase, they're beginning to pull apart slightly. Early telophase, the sister chromatids are being pulled apart and to the opposite poles. And in late telophase, you're finally having the chromatids condense, or the chromosomes condense, and the nucleus is starting to form again. Here's another picture of mitosis, but we have the G2 interphase first, and then we have prophase. Prometaphase is the process of getting to metaphase where the sister chromatids are lining up, but it's after prophase when the chromatin has actually condensed. The pulling apart of the sister chromatids, and then telophase and getting ready for cytokinesis. Here's what it looks like in a plant cell. You've got your interphase, prophase, metaphase, anaphase, telophase. And then you have your cell wall forming right here. Here's another plant cell. Interphase, prophase, prometaphase, metaphase, anaphase, telophase. Here's what it looks like in the root tip of an onion. You can see all the different cells. Most of them are in interphase since that's where cells spend most of their time, but you can see other cells dividing and preparing for mitosis. Here's a sample of what it looks like under a slide. And if you have any questions, please feel free to ask next class.